Welcome and thank you for joining us for today's fireside chat, Scaling Through Uncertainty, where we'll be diving into how biotech startups have continued to push the boundaries of innovation to find success throughout this challenging year, as well as what they're looking forward to and planning for in 2023. I'm your host, Sean McCormack, and I lead our professional services team, as well as industry solution teams here at Benchling. I'm incredibly excited to be joined today by Keith Jarvis, who is VP of Process Chemistry at Velocity Sciences, a company that specializes in creating next-gen precision protein targeting reagents. I'm also joined by Dr. Avi Sheshachalam, who is the VP of Product at Future Fields, a company that utilizes some very exciting, dare I say, futuristic technologies to create sustainable recombinant proteins from fruit flies. Keith, Avi, it's wonderful to have you. Perhaps we could start things off with a longer introduction from each of you about yourselves, your respective organizations, and the awesome science that you and your colleagues do every day. Keith, let's start with you.
Thank you. How you doing? Again, my name is Keith Jarvis. I'm the Vice President of Process Chemistry at Velocity Sciences. It all started for me, a little background from me. I went to grad school here in CU Boulder in Colorado for organic chemistry. And I fell in love with nucleic acid chemistry. And so I've been basically focusing my career on a mix of nucleic acid chemistry, organic chemistry, and chemical engineering. It all started for me at Ribozyme Pharmaceuticals, where I started making RNA enzymes. That company eventually turned into Cerna Therapeutics, where I started making a different form of an oligonucleotide called siRNA. After Cerna Therapeutics, I joined Merck Pharmaceuticals. Then I went to Miragen Therapeutics and I started making microRNA. After Miragen, I got into the gene editing field and I joined Editas Medicine and I started making a different form of an oligonucleotide called guide RNA. And now, since this past January, I've joined Velocity Sciences, where I'm making what we call HPAs, high performance aptamers, or RNA aptamers. So it's been a wonderful journey for me, kind of being a process chemist, an organic chemist, and a chemical engineer, making all these different forms of oligonucleotides. And they all have different applications. So it's really been fascinating. I'm basically using the same chemistry to make these various forms of oligos, but the applications have been from gene editing to RNA enzymes. Now I'm at Velocity Sciences, where, as you said, we're making precision protein targeting reagents. And my main focus, my main responsibility is making RNA aptamers, which are very difficult and very unique forms of an RNA-containing oligonucleotide.
Keith, thanks for that introduction. It's certainly kind of a long and storied career there across, I guess, many different companies, sort of the same field the whole time, but different products. So excited to touch more on that in a little bit. Avi, would you mind introducing yourself?
Sure. My name is Avi Sheshachalam. My background is in cell biology and drug discovery and development. I worked in various different therapeutic spaces in cancers, neurosciences, infectious diseases, vector-borne diseases, so on and so forth throughout my career. And throughout all this time, the curiosity of why science works the way it does was the one that pushed me to get a PhD in cell biology. And eventually, I am now translating all of these experiences to understand how products are created, how to take care of our customers, and what products are helpful in different industries. So that's what I do in my current role as the VP of Product at Future Fields. I help the company decide what products, when to develop, and why we should develop those things. So Future Fields is an insect biotech company co-founded by Matt Anderson-Barron and his partner, Jaleel Anderson-Barron. Matt was actually my PhD colleague in the cell biology department. He worked on fruit flies and cells derived from fruit flies, whereas I worked on immune cells. And his fruit flies would always get into my lab. It was very, very annoying. But eventually, he started using the incubator that I used in the cell culture lab to grow cells, mammalian cells. And I asked him, he said he's trying to grow chicken cells for human consumption, lab-grown meat, which is now termed cellular agriculture. So that was the spark of this program. Eventually, it turned into a completely different program. So originally we were a cell ag company wanting to produce meat, but very quickly we realized that the growth factors that go into these cell cultures are incredibly expensive, almost 90% of its cost. So we pivoted, started using fruit flies as our bioreactors to produce recombinant proteins. And now we have a fledgling program that caters to food and health industries.
All right, so to clarify there, it's fruit flies for protein production, not for human consumption, right, Avi?
That's right, fruit flies for recombinant protein production.
A question for you just on the fruit flies really quick before I go into the first topic. How many fruit flies are we talking? Are these rooms full of flies? Like, are we talking millions of flies? I mean, I can't imagine there's a lot of flies.
That's actually a good question. So we grow fruit flies in a very contained environment. And these are stackable trays, blocks that we call them, production blocks. And within a very small square footage, we can produce several hundred kilograms per day.
Kilograms of fruit flies.
Yes.
Wow.
Biomass per day. Yeah.
That is a lot of flies. Okay, yeah. Well, very exciting. So we have a cell biologist from fruit fly production for protein company. We have Keith, a process chemist. I think the first topic will be pretty exciting here. We're going to talk about sort of innovation in the industry. I have a few primer questions, but we can kind of take it wherever we'd like. Really what innovation is the lifeblood of a startup. Both of your companies are relatively new, they're scaling. I'd love to hear just your take on innovation in biotechnology, maybe specifically how you continue to innovate as you grow, maybe in how you continue to innovate in the tough macroeconomic conditions that we're sort of seeing in the industry today. Let's start with Avi on this one, and then we'll go to Keith.
Sure. I think something changed during the pandemic. I think we can all agree that the power of biology came into its full force with every layperson becoming an overnight expert in mRNA, vaccine technology, and immunology, complex T-cell biology, everything on WhatsApp and Facebook. So something really, really did change. But it also kind of woke up some of the investors and venture capitalists in the space to understand that we as a species face several imminent threats from the climate change impact that we have. And we need to develop technology and solutions to do these things. So I think that is essentially the core of our purpose that we rally around as a team to work on. So we do this using insects. So, Drosophila or fruit flies have been used in academia for over a century as a model organism. And it has been a classical genetics tool. But when we combine this classic insect technology with synthetic biology, now you have a completely different level of game, and it opens up a wide variety of programs. So recombinant technology, protein production technology in general, has been using single-celled organism or mammalian cells of a specific type. But with these insects, we can scale up an entire neuronal tissue, all types of tissues that fruit flies have, at scale, and produce proteins in specific tissue compartments because we know so much about its genetics. So our innovation comes from these kinds of marrying two different fields, but also understanding that the purpose of our company is in the climate change initiative space.
Avi, did you find that investors were more keen to talk about your innovation, about science post-pandemic? Did it sort of change, your sort of perspective on, or their perspective on your company?
I would say that education level on recombinant protein and its ability to change the world has picked up pace over the past few years, and the amount of education you have to give to a finance major has reduced considerably. So in some ways it has helped. In other ways, they're now asking more deeper questions. Can you do mass balance? Things like that. So it's kind of fun.
Yeah. Keith, I'd love to hear your perspective. You said working across kind of the same types of molecules as a process chemist for 20 plus years, of course, different companies. So I'd love to hear your perspective on innovation and maybe also how Velocity itself is innovating.
Definitely. It's really interesting because of the pandemic. Nucleic acid chemistry and all things oligonucleotide kind of blossomed again, thanks to the mRNA vaccines, thanks to Moderna, companies like Moderna and the vaccine, RNA sciences and RNA medicines really got put into the spotlight, and everyone saw how great that innovation was to help create these vaccines. And with that came along all RNA containing oligos. So siRNA really started to blossom, and it already was. And now we're making RNA aptamers in the gene editing space. There's guide RNA. And so a lot of that innovation was already happening. And then when the pandemic hit, it kind of put a spotlight on biotech and RNA medicine. So that kind of was helpful in a way in the very beginning. And now over this past year, it's been supply chain issues have started to creep in, and it's gotten a lot harder. So we were able to establish companies like Velocity Sciences, and we're a proteomics-based company. We're making these RNA aptamers to measure protein binding, and specific to a protein target. And so we were able to we had already done the innovation, and then because of the pandemic and the surge in biotech and RNA medicines, we were able to kind of capitalize that and keep pushing forward. So the innovation was already happening, and then we were able to just continue the research that had started, and there was more interest, I think, because of the pandemic. So it's kind of an interesting case for RNA medicines.
Yeah, so kind of riding this wave of interest. It sounds like you are facing some supply chain issues in 2022. Can you talk a little bit about that and kind of what challenges your company's been facing this year?
Yeah, definitely. Supply chain is a whole different story. So whether we're talking equipment, and it's hard to, if you need to order new equipment, Velocity Sciences is in startup mode. So it's been about almost 2 years now. And of course, that means in startup mode, we're ordering all kinds of equipment, whether it's a HPLC or a mass spec or a DNA RNA synthesizer. And so everything, it used to take 3 months to get a piece of equipment. Now it takes 6 to 12 months. So that has been very challenging. Fortunately for us, I have a lot of connections in this industry because I've been buying these things for 2 decades, as you just described. And so that was helpful, but it's still really hard to get some parts. The computer chip issue, it's definitely been a struggle to get a lot of the equipment in place. So you kind of have to just get smart about it and specialize in the critical equipment and kind of work with what's available. And we've been fortunate that we've been able to do that over the last 2 years. Supply chain, phosphoramidite chemistry, which is required for making oligonucleotides. Phosphoramidites are a main supply chain component, and it's been really hard to get all sorts of reagents that you use for oligonucleotide synthesis. Even like some of the simple things like buffers and synthesis supports have not been in stock. I've been buying DNA phosphoramidite for 20 years now, and it's always been readily available. And even that doesn't arrive as quickly as it used to. So it's been quite challenging, but as long as you kind of specialize and make sure you're using the critical reagents and your critical equipment, then we've been able to get online and push forward. Yep. I think it's going to be that way for years to come. So it's kind of a new era in terms of supply chain for both equipment and reagents.
Avi, I'd love to hear if you're experiencing similar challenges.
Absolutely. I completely reflect what Keith just mentioned. Supply chain has been a big bane during this time, both from the pandemic, but also in the war in Ukraine at the moment. So both have affected us in many ways. Similar to Keith, we kind of worked with what we had. We, leaned down our program, started thinking about what to prioritize. But we also had the good fortune of working in a small ecosystem like Edmonton, Canada, where we can rely on the university or the other partners within this biotech space. And we supported each other during this time, kind of work with both knowledge as well as equipment-wise to use the facilities that exist as opposed to trying to build something from scratch. But as we move outside of the space now, we want to become a bit more ambitious, start having our own proprietary equipment and clean rooms, on and so forth. So yeah, our efforts have been hampered by the supply chain management. At the same time, it has become a space for us to innovate and connect and network with each other and kind of resolve this challenge together as a team. So that has worked well.
You mentioned that you're in Edmonton, Canada, and you're kind of associated with the university in some way. Can you talk a little bit about how Future Fields is scaling right now, what that looks like, what your growth and hiring looks like, and how you maintain innovation in that process?
Yeah, absolutely. So originally, when we started as a young startup, we used the university's startup incubator space to grow, which was suitable for us because we didn't have to buy equipment, a capital-intensive program initially. Fruit flies are incredibly cheap to rear. We use Home Depot blocks to grow them. It is not that expensive, and supply chain on those things are not that hard. They don't eat fruits, they eat yeast, which is the brewer's waste. Edmonton has a great microbrewery scene, so we kind of partner with those people. So in that space, the fruit fly growth is okay, but it's the downstream that we kind of struggle with. But as we grow, we moved into our own facility for our pilot scale, and we're building a clean room facility to meet food and health standards, regulatory compliance-wise. So to do these things, we need to have modular systems and so on and so forth to help us do that. And that's where we are currently at, design and planning and initiating those programs.
Oh, that's fantastic. Pretty funny that you're using brewer's yeast to feed your flies. You can go sample some beer and then feed the flies after.
Exactly.
Good system. Keith, are you growing at velocity? Can you talk about how you think about kind of hiring a culture that's innovative when you scale?
Yes, definitely. When I joined in January of this year, I think I was the maybe 12th employee, and I believe we're at like 16 or 17 right now. So we have been continuing to grow. We basically have a biology department, and we have a chemistry department, and then we have a bioinformatics team as well. And so we've added a few scientist positions to all three of those groups. And it's, we've been growing at the expected rate, I would say. The company started with about 6 to 8 people, then it went to 10 to 12. Now we're at 16 to 17. And I think that was right in alignment with what we projected for 2022. We want to get up to like maybe 25 people over the next couple of years. And I'd say we're right on track basically with our hiring, and we have all the positions covered. When you're in startup mode, everyone kind of has to do everything. Like I might be in the biology lab for a little while. Some of the chemists might go to the biology lab. Everyone really helps out, and we're kind of in that phase. And it's a really fun, that startup mentality is a really fun phase for me. So that's kind of where we're at right now. So we have lots of scientists doing multiple roles and multiple tasks. And that's pretty normal for this phase. And I think that will continue for at least the next year, if not a couple of years.
Awesome. Yeah, I think startup mode is definitely a time where you're wearing a lot of hats. So I'm sure it'll be that way for both your companies for a while. Maybe the next question. Oh, go ahead, Keith.
I'm sorry. Startup mode is so much fun. Like this is my favorite time of being at a biotech company.
Do you share the same opinion, Avi?
Yeah, I like that. I like what he said. So it's true that we do wear a lot of hats. Some days you're a product person, the next day you're a cell culture technician. It's just the nature of the program. But we grew quite fast. Even last year, we grew from around 18 or so to 30 plus people now. And we are growing to 40 within the next few months. As I said, we are building this clean room for manufacturing. So we need to hire experienced manufacturing people to do those things. So we are growing at an incredible pace, but it's the core thing that we have held on to during all of this time is culture, which has been part of our day one of our growth. So that has really helped us, develop this flexible, agile team that has a psychologically safe space where they can fail fast, learn fast, and then, continue to iterate on their programs. And that, I think, has continued to help us as we grow into teams that are much bigger than a startup. As 20 people, it's easy to communicate. At 40, you need structures and frameworks in place so you can communicate in a different way.
And that's going to actually be a perfect segue into the next question, where I want to talk about kind of data and systems and processes that you use to scale. But maybe before we go there, I want to ask one question to each of you. Like, what is the most interesting or surprising startup hat that you wore this year at your respective companies? So what thing did you find yourself doing that you maybe didn't expect to be doing as the VP of process chemistry or the VP of product?
Mhm.
I can
Yeah.
go first. I
Yeah,
would
go ahead,
say
Keith.
for me, it's business development.
Is it like, how do we sell this thing, or?
Or, we're creating these diagnostic toolkits for all sorts of customers. And I'm a hardcore chemist and scientist, and I really enjoy being in the lab. But at this position and in the startup mode, I found myself doing a lot of business development as well, or at least contributing to that part of it. And that was really fun for me, but I was not expecting to do that.
Yeah, absolutely.
I I would say it's the product role itself. Like I didn't know it's such a role existed in a biotech space. I've always been a scientist, helping science progress, but suddenly I had to be at this interface between business and science, working with both spaces, and that has been a learning curve, and I'm really enjoying myself.
And Avi, for you, is that like figuring out what growth factor to produce next, or kind of what the demand is out there in the field, or what is it?
Both, and also merging that with regulatory compliance, talking to the FDA or the CFIA or another group to make sure that we are not going to kill people at the end of the day, and
That's always
maintaining
important.
quality standards, all of these things.
Yeah, absolutely. Okay, well, let's pivot to the next part. The next topic is data. You, of course, are both at scaling organizations. You're both using Benchling, which we can or cannot talk about depending on how you're feeling. But we would definitely love to hear about how you think about building out systems, data systems, processes for your team to communicate, for kind of organizational knowledge to be shared. So let's start with you, Avi, and then we'll go to Keith.
Sure. I came across Benchling, I think, during my grad program. I used it mostly as a DNA repository and trying to score the CRISPR guide RNAs. It's super useful. And I worked with terrible immune cells that hated transfections, and I had like one shot to make them, and scoring them using Benchling really helped, and I actually managed the knockout that I wanted. So that was my first intro to Benchling, and several years down the lane, now here we are in a different setting with a different program, and Benchling has become this massive force in the data management system. So data is at the heart of our company. It's a science company. Naturally, evidence is a proof that the pudding has worked. So we heavily rely on it. I remember when Kayla, the onboarding to help.
Kayla
Yes,
actually implemented both of you and brought you both onto Benchling.
Yeah, and she's fantastic. And I remember, we got on a virtual call with her, with the rest of our team, and they were spitballing right then and there about how to look at the process. And Taylor was just like quietly tapping on her computer and like making this long train of process, like a train, with different compartments, every parameter in the process. And I was just looking at it thinking, this is amazing. And I still go back to that image that she created like almost a year ago to help me realign myself in what process and what step we were at.
Business process is, yeah.
Yeah.
What other systems do you use? Obviously, you have Benchling. How do you guys communicate outside of that? Do you use Slack? Are there other softwares that you use?
Yeah, initially, we use Slack for internal communication. We have an EQMS system that helps us maintain quality and regulatory compliance. We're in the process of getting an ERP that helps us with the product management, inventory, and so on and so forth. So we have a multi-system talking to each other.
Yeah, definitely. And maybe before we go over to Keith for your answer, Kayla works on the implementation team here at Benchling. And, when we implement Benchling, we meet with a different scientific team to try to map out the process and then sort of configure a solution in Benchling that'll serve the need. So for future fields, very exciting because it kind of looked like strain development, but the strains were actually fruit flies and no one had really seen that before. So that was pretty fun. I think Kayla's had a different experience over there at Velocity. So maybe you can talk a little bit about how you think about data management, both with Benchling, with other systems, and with kind of communicating with your teams.
Yeah, definitely. So data management is critical for any type of process, especially oligonucleotide production. I remember back at some of my early companies, everything was paper notebooks, and that was a nightmare. We all know about that. And then at some of my more recent companies, we would have like custom software implemented so we could do, for data management.
Yeah.
And then at my last company, when I was at Editas Medicine, we used Benchling, and I absolutely loved it. And that was my first experience with Benchling. And so when I joined Velocity in January of this year, there was no electronic lab notebook system, of course, since we're in startup mode. And Velocity has an extraordinary amount of data to manage, whether it's from the bioinformatics team or from selections in biology, or whether it's from oligonucleotide production or any of those processes. And so, you know, I told everyone here at Velocity about my great experience using Benchling at my last company, and we really should consider using that here. And, you know, we did a little demo, and we got to meet Kayla, and the rest was history. It was a no-brainer to sign up for the startup mode for Benchling. It's so important if you're making an oligonucleotide, whether it's being used as a diagnostic or a therapeutic, that you track all the data associated with that batch, that lot, the quality, the analytics behind it, all of that, the lot numbers of the reagents, all of that needs to go into some type of inventory data management system, and Benchling is really perfect for that. And then it goes way beyond just, you know, documenting that data, but you can do process development projects and document all of the process changes and details that go along with like a process development project. And then there's a quality component to it as well. And I think that is something that we will even expand into in the future. So it's critical that you have a good system that manages your data and makes it easy to show that data to someone else. Like as a new employee at a company, you want to come in and be able to study what everyone's been doing, the outcome of their process development projects, a research experiment, whatever. And then you can go into Benchling and all that data is there, you can search. It's just awesome. And so it was very easy for me to get the whole Velocity team on board to implementing Benchling. And actually, we hope to go live January 1st. And so we're super excited. We've been working on the implementation phase for months now. And yeah, it's going to make our lives so much easier for next year.
I know Kayla's very excited. So I guess there's just like this general idea that if you put all of your data into a system, you know, from start to finish through your experimental pipeline, that you might be able to ask, you know, important questions, improve your processes, like dig into insights that you wouldn't otherwise have done with. I want to know, Avi, if that's your experience, whether it's Benchling or it's elsewhere, like how are you using kind of the insights that you're gathering from your data set to improve your processes, to improve future fields outcomes?
Oh, that's a great question. Absolutely. Data management makes such an impact on knowing where we're heading. So both internally speaking, so, you know, having this process mapped out and starting to put data in and then visualizing that data as the experiments move on. That is one part of R&D that we needed and now we have in place. The second part is process development. This is where things like repeatable processes come into place. This is where the templates can start taking shape. And eventually this translates into manufacturing setting where the templates become the de facto way to input data and making sure that it's now compliant with a regulatory body that can come and audit us and go, okay, I click on this and goes all the way to the gene and, you know, have this one track of information in one place. So absolutely, the data management is a key element of making sure that the products are safe, or that we are able to produce quality products in the process, and that we are able to track its development, yield, titers, so on and so forth in the process.
Do you have data scientists at the Future Fields right now that are kind of asking questions of your data set?
Yes, we do. We have a couple of engineers who help us, you know, manage this. Yeah, it's a behemoth of a project. So I'm very glad that they're here.
Yeah. And what about you, Keith? What does kind of asking questions of data look like to you once you have it all in a system?
Wow. So it's kind of varied. You know, there's data that come from like assays, chemical and biological assays that, you know, we use for quality and purity measurements. And so we need to access and store that data and make it available to anyone who might be using like the oligo that was made. What are the analytics around this that I'm going to use for my next biology experiment, for example? Mm-hmm. And, you know, we have a bioinformatics team that's like generating new sequences, tens of thousands of new sequences. Those all need to be managed and tracked. And we need to store data from biological experiments, and we need to store data from, you know, process optimization and process development experiments as well. So all of that data we're using Benchling to kind of store all of that data, and it's critical. It's critical as we scale up as well, and it will enable us to scale faster, I think, like increase throughput.
Maybe a question now is thinking sort of the future of both your companies. I know the next topic is next year and what you're thinking there, but I want to talk about sort of how you envision managing your data in the years to come, how you envision your company scaling with the infrastructure that you have. And so maybe, Avi, if you could start with that question and just think about, where do you want to be at Future Fields in terms of how you're inputting data, how your scientists are interfacing sort of with that data set?
Yeah, so that's a great question. So we have several types of data going into the system. So we have the insect stock maintenance data, but we also have a synthetic biology component to it, the genetics part of it. There's a protein component, which is the product that we produce, but there's also the cell component, which is the validation part. So we have very different type of data going into this one system. And we need to maintain a certain level of quality in amongst all of this. So as I mentioned, we go from R&D to process development to manufacturing. And during this time, the way I envision is that as the products enter into the process development phase and towards the manufacturing phases, we want to have a template that helps, that reduces the burden on the manufacturing people, but understands that there are critical elements of a particular program that are already, in a particular format. All you need to do is enter, specific numbers in that space. So this helps the whole program and the whole company streamline its process. So this is how I hope the data management system focuses our research.
So maybe to say that back to you, you have groups that are kind of growing the fruit flies and the fly stocks. You have groups that are sort of developing the sequences that will be turned into the proteins that the flies are producing. You're doing protein production and purification. And then ultimately you want to manufacture that at scale. And you want to do this all kind of in the Benchling system.
Yes.
Wow. Okay. That's fantastic. I think that's our dream as well. So it's exciting to hear that sort of Future Fields shares our vision. Keith, maybe can we talk to you about sort of where you see the future of kind of a scientist's interaction with the data system going, where you want to be at Velocity next year or in the years to come in terms of that kind of a viewpoint?
Right. So we are not only making RNA aptamers, but we're also making various other forms of oligonucleotides. And so it's critical that we're able to track all of this data, and that we eventually move to a quality management system. And I know there's a quality component to Benchling that could be in the future for us as well. So I think that as we continue to scale and also increase throughput, we'll want to implement a quality management system. We'll need to be able to track the metrics and the quality of every oligonucleotide that we produce. And we'll need to have like a quality management system, and hopefully we can implement the one that's available through Benchling. So I think that's where we're going to go over the next couple of years. And we need that whole history, the whole history of the process, everything, all production. And then how are those oligonucleotides or RNA aptamers used? And what is the outcome of those experiments with all these different variants of our aptamers, for example? So we'll track all the data, the purity data, and we'll track the results through its application. And all of that will be done under a quality management system. That's kind of like our long-term vision for Velocity Sciences.
So it seems like kind of amongst both your organizations, there's a desire to be on fewer systems as opposed to more. I think, is that a fair statement?
Yes.
Absolutely.
I think we have faced the same, we have the same issues here at Benchling. When you're implementing software, you want to be on fewer systems. You want to interact with fewer systems in your day-to-day. Cool. Well, let's talk a little bit about next year. In 2023 is really just around the corner. We're in the middle of December, I guess, early December. What does next year look like for both your organizations as you continue to scale? What are you excited about? What are you excited about in the industry? I suppose you're probably not excited about the continued supply chain difficulties, but there's got to be some things that you're looking forward to. So Keith, maybe to start with you, and then Avi after that.
So what am I looking forward to next year? I'm looking forward to having Benchling active on January 1st and getting through the implementation phase. It's going to make everyone's life so much easier having this electronic lab notebook system and having everything. Just being able to, it's going to make their daily process better and their daily routine better. but the sharing of information is what is going to be so great. We'll be able to, you can go in and look at someone else's entries and you will be able to share data that way. And I think it's just going to increase communication and make us more productive. And, I'm really looking forward to having that capability and then scaling up, increasing our throughput of oligo production, increasing our selection process, documenting everything that bioinformatics is generating, and then be able to share all that data. That's, it's going to be awesome next year and the year after as that all comes together.
Yeah, I guess you're speaking from experience here because you kind of had the experience of all of this at Editas already, right? So it's not
Exactly
pie in the sky here. It's actually been validated. Avi, what are you excited about for next year? What is Future Fields looking forward to? What are you looking forward to in growing your organization next year?
More products would be really nice. So new product development, that's what I'm looking forward to. But as I mentioned, we are moving towards manufacturing. That would be a really exciting step for us, scaling up and really becoming a biomanufacturing facility in Edmonton, providing this sustainable recombinant proteins, not just for food applications like cellular agriculture, but also in the health spaces. Our appetite is quite big. We have much bigger eyes than we can chew. So we want to continue to develop in that space and, have this ambition driving us. But to be perfectly honest, I think it's the team that helps us do these things. They're so flexible and brilliant people and rallying around this singular purpose of making sustainable recombinant proteins. That is what we are all hoping to achieve, not just this upcoming new year, but also in the years to come.
That's super exciting. I guess as you move into manufacturing, does the scale of course changes? Are you still using sort of the fly trays or will it be different?
Yes, the fly trays are really stackable. They can go all the way to the ceiling. They're incredibly easy to maintain. Yeah. They are going to be in those trays, production blocks, we call them, and we will produce several 100 kilograms a day and then process them.
What do you do with all the waste material?
That gets processed in different ways for byproducts. So proteins, lipids, chitins, so on and so forth that come out of insects have multiple applications. And we have a zero waste biomanufacturing plan in place. So we'll see where we can go.
Awesome. Well, very exciting conversation for both of you. Anything else that you'd like to kind of close with about your excitement about next year, about things you're looking forward to, maybe just a take on the biotech industry generally?
Sure, I can go first. So I'm super excited, like I said earlier, about Velocity Sciences and meeting our customers' needs over the next couple of years. It's really fun to be in this startup mode. This is the most fun I ever have in biotech, is when a company is just starting and you really get to drive it and create that business or that therapeutic or that diagnostic toolkit or whatever you're doing. I'm really looking forward to seeing that come to fruition and seeing all the hard work pay off and becoming a mid-sized company and getting out of that startup mode.
Yeah, absolutely. What about for you, Avi?
Well, I really like the purpose of this company. I think that's what I want to continue to do: produce sustainable, effective recombinant proteins that help, resolve some of our questions on food insecurity in the world, or with respect to health, where only effective proteins are the norm. There's no talk of sustainability in health applications. So it'll be nice to bring those elements into different markets.
Yeah,
that's
absolutely.
what I'm really excited for.
Well, the Benchling team is certainly rooting for both of your companies, for both of you. Avi, Keith, thank you so much. To everyone that's tuning in, thank you all again for tuning in today. I hope you learned as much as I did from the insights Avi and Keith shared. If you'd like to learn more about investing in data management and how that can help your biotech find success in the new year, please vote on the poll on the right-hand side of the screen. And then Avi and Keith and I are going to stick around and do some Q&A over the next 15 minutes or so. So please feel free to put some questions in the chat and we'll take those questions. Thanks to everyone. Awesome. All right. Now time for a little bit of Q&A with Avi and Keith. If you have any more questions, for those that are watching, you can just put them in the Q&A sidebar in the chat there. Maybe we could start with a question for both of you. We'll start with Avi, just about the unique challenges you face building a biotech company in Alberta, not in a major biotech hub, of course, close to the university, but if you could start there, then Keith, maybe same question to you after about building a biotech in Boulder.
Sure, I can start. Well, as you said, biotech is quite a new phenomena here in Edmonton. Over the last few years, small startups have started in this space, and they're relying heavily on the university, this massive program that spits out talented technicians, research scientists, and postdocs, and entrepreneurs from the space. And they all often go to the US, brain drain from Canada. But now we have an opportunity here to diversify Edmonton's economy from its usual oil and gas industries towards the sustainable clean energy as well as biotech spaces. So that's our uniqueness of staying here. Challenge-wise, well, it's pretty cold here. I think Sean and I were just talking. It's minus 40 this week, degrees centigrade, and it's really hard to attract people from San Francisco or Berkeley to come in and enjoy the cold here. But we're close to the mountains, so there's other attractive features here.
Yeah, 40 degrees below is pretty cold. I guess no one's leaving the lab, so you get more time in the office. And maybe the flies are flying a little slower.
That's true.
Keith, how about for you over in Boulder?
It's kind of the opposite here in beautiful, sunny Boulder, Colorado. And actually, there's a lot of biotech in this area. So I would say it's easier to build a biotech company in this area. A lot of you might know that nucleic acid chemistry and all things oligonucleotide kind of started in this area with Marv Carruthers at CU Boulder. We kind of consider him the godfather of nucleic acid chemistry, and he, a lot of companies have started and remain in this Boulder area. So there's a lot of biotech that's already been established. There's, phosphoramidite chemistry does not work very well with humid environments. So we have a very dry environment out here in Colorado. So that's helpful as well. And it's a great place to start a biotech company. And there's a lot of biotech in this local area, a lot of talent. So it actually makes it easy. And we have over 300 days of sunshine a year. And then, of course, we can drive to go skiing in about an hour and a half as well. So it's a fantastic place to work and live and play.
That sounded a bit like a sales pitch, Keith. If anybody wants to work at Velocity, you can reach out to Keith. Maybe the next question, well, we can start with Keith. How do you prioritize data collection and kind of establishing those data collection processes when there's like little to no infrastructure in place at your scaling company? So how do you kind of work with those two parameters?
I see that question is from Michael. Hi, Michael. I've worked with Michael in the past. It's great to see you online. So how do we prioritize data collection and the processes? So that is a great question, and it is a big challenge, especially in startup mode. It's really easy for me because I'm in charge of oligonucleotide production. So I've been doing this for 20 years. So I know what data is most important to produce an oligo. I know the quality standards that go along with it. And so as we create our own personalized bench lean, I know exactly what data I need collected, and I know what is the most important. So since I've been doing this for so long, it's been really easy for me to create that priority. It's really all about process observations and the analytical outcomes of the final product when you're making an oligonucleotide or an RNA aptamer in the case of Velocity Sciences.
Keith, is it fair to say that you know what initial data must be collected, and then as your team grows and scales, you start plugging in additional assays, or do you want to have it all built from the get-go?
Well, it's always going to have to evolve, but we're trying to build it all in right in the beginning. As you know, it's been great working with Kayla, and we're really pushing forward to get everything as much as possible implemented before we go live next year. Having said that, we're using some novel chemistries, and we have some very novel ideas, and we'll have very novel products. So I'm sure the system will have to evolve over time as we continue to develop those products.
Yeah. Okay, Avi, same question to you. Thinking about how you prioritize data collection when there's little to no infrastructure in place at your scaling company.
Yeah, we started with one person in the lab. That's how the startup worked. One person writing down things in their notebook and, trying to make genetics happen on fruit flies. Eventually, with 10 people, we prioritized on hiring our quality person. And this quality is at the heart of our processes. And eventually that's the central focus for data collection. This is why we transitioned from notebooks to electronic notebooks towards Benchling that is compliant in a specific space. And this evolution is mostly driven by what we have planned for the future and bringing this Benchling and other compliance softwares very early on in our processes to ensure that what we can do in the next year, we are already within that space, working with that space.
Great. Awesome. So maybe next question. I want to know if either of you can actually speak to this, but a little bit about fundraising and biotech today. I don't know if either Velocity or Future Fields are currently fundraising or thinking about it, but I would love to get a take on the environment now and what you're thinking about. Maybe Avi, for you first.
Sure. Yeah. I mean, as a startup, we're always fundraising. So please hit up Matt at Future Fields if you're interested in, talking about money. But yeah, so fundraising it's been an interesting experience for us. What we could raise as a seed grant in Edmonton, Canada, versus what it is usually in the Silicon Valley or in New York or so on and so forth in these biotech hubs is very different. To start with. So we had to learn to be scrappy. We had to learn how to utilize the facilities that we have access to and network and find partners who can work with us and leverage these networks. So that's what we've always dealt with and we've always managed. But the war as well as pandemic has changed in some ways what the fundraising scenario looks like. And because we were so scrappy to start with, we worked with what we could get. So we are about to close our fundraising cycle, and we will have information on that out soon. But in the process, we planning, technical due diligence, so on and so forth. All of these things matter quite a bit, and having these things ready for people in the finance sector really matters now. And this plan really persuades them to invest in good companies.
Do you think that Future Fields is a stronger company because of how you raise money and because you had to be scrappier for longer before kind of getting that first big check?
I think so. I mean, we leaned on our program. We had, as I said, we were very scrappy about how we spend every penny. And by doing this, we've gotten really good at how we can get the best bang for our buck.
Yeah, absolutely. Keith, how about to you? Same question.
Yeah, I agree with everything that Avi just said. It was really easy. Fundraising was actually easy in 2021, and it became much harder in this year in 2022. Having said that, fortunately, Velocity was established in 2020. So at the end of 2020, or beginning of 2021. So it's definitely, the biotech landscape has definitely changed along with the global economy. So it's become a lot harder, but I think as long as you have a plan in place, and I feel like for a biotech startup, you always have a 2-year plan, right? And we have that 2-year plan in place. And as long as you really do a great job of planning and continue to innovate and create those novel new products, then the fundraising, becomes much easier, even in tough times like this.
Yeah. Maybe one more question sort of on that planning. You mentioned a 2-year plan. What does that plan look like as you're shaping it at your company, Keith? And same question to you in a minute, Avi. Are you planning to develop a new product? Are you planning to scale that product? How do you sort of go through those planning cycles?
Yeah, that's a great question. So for the last year and a half, let's say, we've really been in the implementation phase. So ordering the equipment, getting it online, getting all of our assays developed, getting our processes developed. Again, we're making these things called, these RNA aptamers called HPAs, high performance aptamers. And we're moving toward having a product and supplying that product to our customers. We have a list of potential customers now, and our 2-year plan really is to execute on the startup phase and then start delivering to our customers.
Yeah. Okay. Absolutely. How about for you, Avi?
Yeah, that's a good question. So So I think because we are a recombinant protein company and we were trying to serve cell ag almost exclusively earlier on, we needed to quickly diversify these market spaces. We needed to look at other markets that can also use recombinant proteins that we produce. So that's one way of risk mitigation for us. Another aspect is in the same vein is that the manufacturing schedule, the facilities that we're designing, and the people we are hiring are all accurately in line with these big plans and diversification of the markets that we have. And if we cannot do those plans in the exact way we have, it becomes harder to persuade people to invest in the company. So we went through these cycles, we went through these planning phases to explicitly explain our program to different people, to different stakeholders.
Yeah. Avi, is there a limit to what you could use Drosophila for in terms of making recombinant proteins? I mean, it's currently cell agriculture. Could they be clinical therapeutics someday? Like, what is the limit?
Yeah, that's what we want to do. We want to make humanized proteins from insects. Insect cell proteins are currently used in the therapeutic spaces. So why not insects? They can make evolutionarily, they're much closer to mammalian systems than single-celled organisms like yeast or bacteria. So they make proteins far, far better suited to the mammalian systems. So why not?
Well, yeah, no,
Yeah.
I don't know. I don't know why not. We're looking forward to it. One kind of meta question here is about sample labeling, but maybe we can expand this question and talk about like how you manage inventory in your facilities. How do you keep track of things? How do you label your samples?
I can go first. We have a quality assurance person who helps us with it. They have very strict protocols on how these things happen, and product validation leads them towards this quality control process, and then eventually we have a labeling mechanism for our products. And inventory management system is currently in its infancy for us, let's say. So we are using easy accessible solutions for this, but eventually as we have more products and several vials of each product in different sizes, we will need an inventory management system like an ERP.
You say easily accessible solutions. When I was in the lab, that meant like an Excel sheet and a Sharpie.
Oh, it's not that bad. It's very close to what we use.
How about for you guys?
Pretty much the same answer right now. Since we're in startup mode, we're using, we don't handwrite labels, of course, but once we have Benchling online, actually, we're going to have IDs that go with all of our products, along with lot numbers and batch numbers as well. Right now, and maybe barcodes is in our future. We'll see how that goes. Right now, we're just using cryo labels, and we're using a printer to print those labels. That will definitely evolve next year, especially once Benchling is up and running, and we'll definitely streamline that process.
Yeah, definitely important to kind of label everything in the -80. But Avi, I feel like you guys can just put your stuff outside.
Not really. I mean, well, the final product is lyophilized, so they're actually quite stable at room temperature. So we ship our products in an envelope, Sorry, yeah, that's true. That's also true.
Great. Well, that's all the questions we have in the chat. Avi, Keith, did you have anything else you wanted to touch on before we go?
No, this has been a pleasure talking to you and learning from both you and Keith. And yeah, really looking forward to using Benchling and all its avatars in the future.
Awesome. Well, we certainly appreciate you both. Thank you very much.
Yeah, I echo that. It's been great. Thanks for having me. It's been an absolute pleasure developing Benchling. We're super excited to get Benchling online. So thank you for that.
Awesome, Keith. Thank you so much. Great. Well, thanks everyone for joining today. Tune in next time for our next Benchling webinar. Thanks so much.